Good day, Honors Biology students. Welcome to video lecture episode 20. Today we begin section 2 of chapter 12. Remember, chapter 12 is titled Molecular Genetics. Well, section 2, now that we're getting into, is titled Replication of DNA. So let's talk about the two objectives for section 2, and they are as follows. 1. Summarize the role of the enzymes involved in the replication of DNA, and the other objective is explain how leading and lagging strands are synthesized differently. Now let's consider the main idea that's going to cover the whole of section 2, and that is this. DNA replicates by making a strand that is complementary to the original strand. Now if you look on slide 4 there, you'll be able to see an image of this, what's represented. Alright? Now let's go ahead and connect this to life outside the classroom, because we always need to see that the information we learn is not just simply academic and we just learn it so we have knowledge, but we can really use it. Now, when one uses a copier machine to make copies, photocopies that is, they intend for them to be the exact thing. In other words, when myself and other teachers, when we've made copies for our classrooms, when we take it to the copier, we intend that we put whatever the document is, the copy, that we want it to be the same, because it's for all the students. We want to make sure they have the same thing. What we intend on doing, and what it's also called, this is also called a carbon copy. In other words, it's the same copy. All right? Now, if when making a copy there were errors on it that were not on the original, it would make copying inefficient and not effective. In other words, if we copied it and it actually messed it up or didn't include things, we'd be like, this is not working. This is inefficient, means it, and it's not effective. It's not doing the intended purpose. Now, in this section, section 2 of chapter 12, we will consider this concept of making copies as we discuss, of course, the replication of DNA. So let's talk about semi-conservative replication. Let's talk a little bit about some people we talked about last I believe it was last lecture, but also in last section, and that was Watson and Crick. Remember, in 1953, they published a paper on the structure of DNA. Well, in that, they proposed their model of how the DNA replicated, and that model they proposed was called semi-conservative replication. Now, what is semi-conservative replication? Well, it is the process in which DNA is replicated so that in the end, each new strand of DNA is an exact copy. Now, to accomplish, they have to have an original DNA strand using a template. So basically what happens is, just like we as teachers, when we make copies of things, we have this original document that we lay down and then we get copies of that. Well, the DNA does that as well. It takes the original DNA and it copies it so that then when that DNA is passed on, all the genetic information is passed there. Now, a template is just something used as an original to prepare for making copies. Now, once it splits, that being the original strand, each of the halves are used as what we call template or parent strand. Then it becomes its own DNA through multiple enzymes and assembling of base pairs. Now we're going to describe this process more in our next few lectures. I do hope this has been helpful. If you have any questions, please leave it in the comments below. Reach out to me via email or message, and I hope once again it's been helpful. Have a nice day, students. Take care.